So we want to talk about mutation now, because ultimately mutation is going to be defined as a change in the genetic material of a cell or a virus, but we don't really care about that other than the fact that that is the information to build a protein. And when we change that information, we can potentially change the protein. And because proteins do stuff, now they're going to do stuff differently, better, or worse, or not at all, or about the same. What we want to look at in this talk at our understanding of molecular biology now, we're going to talk about point mutations. Later, we'll talk about gross mutations, which are huge chunks of chromosomes that you rearrange or delete or add on. But for now, we're going to be looking at small changes in the DNA, just one base pair, or accumulation of a handful of base pair changes. So we've already talked about hemoglobin and how sickle cell anemia is the result of a mutation in hemoglobin. And here, in this situation, changing a single nucleotide changes the amino acid in hemoglobin from glutamine to valine, which causes a sickle cell to occur. All right, so here, a point mutation at just the right location has changed the phenotype, literally, of the cell to result in this disease. Now, not every point mutation is going to have that effect. Right? You can take a brick out of a wall and the wall still stands. And in many cases, that's fine. But if you're looking at something like an arch and you take the keystone out, boom, the whole thing collapses. And so there are certain amino acids that are more important to the structure and function of a protein than others. Right? So there are two major types of point mutations that we're going to consider, and those are base pair substitutions and base pair insertions or deletions. So the substitution is going to be the changing. These are the ones you see on the left side. These are going to be changing a nucleotide to a different nucleotide. And an insertion or deletion is just that. You're going to either remove a nucleotide from the sequence or you're going to add an additional one in. Now, this cartoon is kind of the standard for kind of showing what we're doing when we change things around. And what you'll see at the top of the screen is what's called the wild type. Now, the wild type, when we talk about genetics, is the most common variant of a gene in a population. So just like not everybody in the world has the same eye color, different eye colors result from different proteins. Those proteins are encoded by genes. We have different versions of that gene, and we'll talk about that later, called alleles. Well, whatever the most common allele or version of a gene in a population, we call it the wild type. So for now, we'll call that quote-unquote normal, even though that's kind of a misnomer. Now, when we change that, then we have mutants, right? So again, we have base pair substitutions or insertions and deletions, and what we want to do is look at each of these in turn. So with a substitution, we're going to replace one nucleotide with a different nucleotide. So silent mutations are going to be the change in the nucleotide sequence that plays upon the redundancy of the genetic code. It's degenerate. So I can actually change the nucleotide sequence, but if I get a codon that encodes for the same amino acid, the protein's the same, we call that a silent mutation. Missense mutations are going to code for a different amino acid, and nonsense mutations are going to result in a stop codon. So if we look at this, a silent mutation here, again, so we see our DNA up at the top, we see the resulting mRNA, and from here, when we change that G to an A, you actually get the same amino acid there, because GGC encodes for glycine, as does GGU. So we call this a silent mutation. There's definitely a change in the DNA, and that's our definition of mutation, but we haven't affected the protein, not the amino acid sequence. Now, a missense mutation is going to change the amino acid itself, right? So you've got a new series of amino acids. So here, instead of a C, we put a T, and what we get is a serine instead of a glycine. So GGC encodes for glycine, but AGC encodes for serine. And now, that may or may not affect the protein, right? And that depends on the importance of that particular amino acid in the overall three-dimensional or tertiary structure of the protein. Now, nonsense mutations usually have a pretty big effect because they're the insertion of a stop codon. So here we've changed AAG up top in the wild type mRNA to UAG. So U are gross. And that results in information that says we're done. And so if I'm not going to ever really get to the elongation stage of translation because I've got a stop codon right away, I don't have a protein. So in that case, you've got machinery that says we're cranking out mRNA. This is great. We've turned this gene on, but we're not getting any protein from it, and that's going to have an effect on the cell. Okay, sometimes that's bad. Sometimes that's good. Depends on the protein. Depends on the situation. Now, our other category of point mutations are insertions and deletions. And the other way to categorize these are called frame shift mutations, because this is really going to test your understanding of what we know as reading frame. So we know the genetic code is triplicate, so it's always 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. And every 1, 2, 3 is a codon, right? If I actually remove a nucleotide or insert a nucleotide, now I've shifted the bracketed 1, 2, 3. So in this case, we see the wild type. We go TAC. Let's go with the mRNA. AUG, AAG, UUU, GGC, UAA. Now, if I insert an extra A after that first codon, UAG, now I go UAA instead of AAG. The AAG is still there, but everything's shifted over. And because I only read in groups of 3, the machinery doesn't know that it should skip over or that U has been extra added. It has to go AUG, UAA. That's what it reads as the next 3. Okay? That happens to be a nonsense mutation in this case. All right? But you've shifted everything. So if we look at the next example, extensive missense based on a frame shift, we can see here we've inserted a U into the mutant mRNA there at the bottom. So at the top, in the wild type, we go AUG, AAG, UUU, GGC, and then a stop. Here we go AUG, AAG, and then instead of UUU, we're going to delete that U, and everything shifts over one. So now we go UUG. That G used to be the first nucleotide in the glycine codon, and now it's the last nucleotide in the previous codon, and now you get leucine instead of phenylalanine. Now, not only have you changed the phenylalanine to leucine, but you've shifted everything over, and now the next codon to read, instead of being GGC, is GCU. And alanine is encoded. And now you've erased, in essence, your stop codon. It's fallen into a different frame. And so this now can go on adding amino acids until it hits a random stop codon in the nucleotide sequence. Now that's going to be a protein that probably is going to be a mess. All right? So frame shift mutations then are the insertion or deletion of a nucleotide, and that can have far-ranging effects on the protein. All right? Does that make sense? Okay. So additionally, you can cluster frame shift mutations. So if you were to get rid of 3 nucleotides in a row, you're going to maintain the frame, but you've lost a whole amino acid. So in this case, you've removed the TTC. Now, you can also see this as you get an insertion or a deletion, and you may get another, let's say go with deletions. You get a deletion somewhere near the beginning of the protein, and let's say 5 nucleotides down, you get another deletion, and then another 10 nucleotides down, you get another deletion. Well, because those 3 are cumulative, you're going to be back in the original reading frame, and now have entered in, maybe 5 or 6 amino acids that are different from their original. If there's not a stop codon on there, that protein is going to just charge on and have that kind of segment that's mutated. So again, the power of the frame shift is pretty significant. So now what causes mutations? Mutagens. And this is something that, again, is a huge topic, and we'll discuss it a little bit here and there as we go through the quarter. But basically, there's always a background level of mutation occurring. So during DNA replication, during recombination, which we haven't talked about, but we will, and repair, errors happen. And sometimes repairs don't work. And that can lead to changes in the DNA sequence. Now, exposure of DNA to mutagens, right? Physical or chemical agents that can change the DNA, those also can result in mutation. So physical agents are going to be electromagnetic radiation. So X-rays, gamma rays, UV light. All those can damage the DNA in such a way that it can change the sequence. Or chemical agents, all kinds, classes and classes of chemicals that can interact with the DNA and damage it or change it.